All right, hello and welcome to this session. We will start the talk shortly, but first a few housekeeping items. If you have questions, please enter them into the Q&A tab. You also have ability to upvote the question. And if you would like to ask your question live, please use the raise hand feature and you can join the stage and ask questions live. There are a variety of emoji available to give feedback, and please use them freely. And if for some reason you have to leave early, video of this session will be available a couple of hours after the session has completed. And now let me introduce you to our speaker, Pacome Prompsy from Institut Curie, and he will be talking about interactive analysis of single-cell epigenomic landscapes with ChromSCape. Pacome, take it away.
Thank you very much, Mikhail. I'm very pleased to be here, and I will start sharing my screen. So, hopefully you can see my presentation. Yes. So, hello everyone. My name is Pacome, and I'm from the Valo Lab in Institut Curie. And today I will present a ChromSCape, which is a bioconductor package and a Shiny application to analyze single-cell epigenomic datasets. So let's dive right in and give you the plan for today. So I will give you a bit of context about single-cell epigenomics. Then I will try to show you a bit of the challenges and how we can analyze such datasets with ChromSCape. And I will present a short video. Finally, I will focus on some of the features that I won't be mentioning in the two first parts, and end with two use cases in which we use this application to analyze single-cell epigenomic datasets. So, for the context. In our lab, we are interested in epigenomics, which is the study of epigenetic modifications that regulates expression. And this can be either openness of chromatin, or DNA methylation, or histone modifications, which we are particularly interested in. And we are particularly interested in seeing those modifications with the angle of single cell, which is the ability to capture and sequence cells one by one, as depicted by this graph, and to uncover the heterogeneity of complex tissues. And there was quite a lot of single-cell epigenomic technologies that came out in the last 5 years. And I've retranscribed this as a timeline, particularly focusing on single-cell histone modifications. And you can see that there is quite an explosion in the two last years of such technologies. And for example, with now a high-throughput single-cell chip-seq or a single-cell cut and tag, which allows to assess single-cell histone modifications in single cell, and per tag, which also embed expression in the same cells as histone modification, or spatial cut and tag, which adds the spatial information and was pre-published on bioRxiv this year. So, with this boom of technologies, there was multiple tools that were developed, particularly for single-cell ATAC-seq. And single-cell ATAC-seq is the ability to assess openness of chromatin in single cell. And in this paper by Chen and colleague in 2019, they assessed several of these packages, and now new packages have been published. However, there was no tools to analyze both single-cell ATAC-seq and other single-cell epigenomic datasets. That's why we wanted to create an application, a Shiny application, to analyze such kind of datasets and could analyze various datasets. And ChromSCape is an application that takes the user from quality control and feature counting towards interpretation, and visualizing the data at each step. And of course, any bug can be a report on the GitHub page. So now that I've given you a bit of background, I would like to dive a bit more into the challenges that awaits you when analyzing such kind of complex datasets. So for RNA-seq and expression, there is a natural feature, which is the gene or the transcript. And when you analyze such data, you would naturally create a gene by cell matrix. However, for single-cell epigenomics, there is no natural feature. That is said, you don't really know how to create your matrix. But we can analyze some features that we can count or read on. And there are genomic bins, which is dividing the genome into equally sized bins. Peaks, which is trying to find signal enrichment in the tracks, and gene, which could be similar for RNA-seq, but on DNA. And actually, there is no consensus on what feature to choose, and ChromSCape allows to count on any of these features. And I want to stress that there is a trade-off that the user has to take. And for instance, if you use picoting, you would pickle on the cumulative signal, which is represented here in red, and is the aggregate of the single reads that are lines here. And you would focus only on regions which are strongly enriched in signal. So this is a more supervised approach that is interesting, but you might miss a lot of reads that falls outside of these peaks, and you might also decrease the overall coverage. So you could try to use genomic bins, which would take the entire genome, but will be more noisy. And this is a balance really between bias and noise. But if we use genomic bins, we have to wonder what size of bins we have to take. And this is a trade-off between signal and resolution. Because if you look at this large-sized bin, you can see that if we select a large bin, there will be a lot of signal in each of the bins, which might be good for the analysis. However, we decrease the resolution as the information that, for instance, 2 peaks are present in such bins is lost. So if you select once again smaller bins, you would have more resolution but less signal. And this is really dependent on your dataset, whether you analyze single-cell ataxic or single-cell cretin type, for instance. You might have really different coverages. So, and now I want to give you an overview of what is ChromSCape with a video. So, if we can launch the video, this will be perfect. So, yes, so this describes how you would use the application. So you simply have to load the application and use the launch app function to launch it. So you arrive on the main page, and I'm showing here that you can simply go to the help buttons to give you a bit of guidance through the application. And the first thing we need to do is to select a directory where we would create our application or analysis. And all the analyses would be stored in this directory. We now create an analysis. I take example of breast cancer using single-cell H3K27 methylation, and select the reference genome, And as you can see on this box, there are several file formats that are available, either matrices or raw file formats, which we are able to count using ChromSCape on several features that I described earlier. We will go on and select our dataset, and here again a little help of the dataset. Select the folder where each of the data sample is, and create our analyses. And you can unselect samples if you want. So we move on to the second tab, which is the filter and normalize tab. In this tab, you would move the sliders to simply select which thresholds you want to filter your data with. And here you want to filter out doublet cells and lowly covered cells. And also take a certain amount of features for your analysis. There are two normalization methods available. You can perform subsampling if you have unequal cells in your samples, and exclude some specific regions, for instance, if you have copy number alterations, and integrate multiple batches using the perform batch correction and the fast MNN batch correction. So here we will simply launch the classic analysis. This performs PCA and dimensionality reduction, and the dataset is available on the right panel. We can move on now to the visualization panel. And this, in the left side is the PCA, and the right side is the UMAP. You can also calculate t-SNE optionally. And here I'm showing that you can change the parameters of visualization for the entire session of the application, and also change the parallel computing parameters. So here we switch back to the labels, and we can change the color, for instance. And this will be saved once you close the application and relaunch it. So if you focus on the two plots down, these are two plots that shows the contribution of features to the first PCA and their location in the genome. And we can see here that in the 100 top features, most of them are on chromosome 7. So we now move to the clustering panel, which is based on Pearson correlation as a distance matrix. And we can use consensus Yachakel clustering for robust clustering, and filter out lowly correlated cells. And this is an additional filtering which helps for a more robust clustering using hierarchical clustering. We see here that we have four nice squares, which are four homogeneous populations. So we will select 4 clusters. And now we have our UMAP colored by the clusters, and we can see that the correlation heat map quite matches the UMAP. One interesting plots that we can create are the intra or intercorrelation plots, which shows the homogeneity of each labels or each clusters. And we can add the single cell information for each of the clusters. So now that we define those clusters, we might want to go a bit further and try to create the coverage tracks for each of the clusters as pseudobulk. So we created the big-wig files using the application, and now we are plotting it on the application to show the difference of raw signal of coverage at some loci or some interesting genes. And here, for instance, we can see that the third cluster has quite an enrichment of the repressive mark as at the beginning of this region. and we can save the plot in high-quality PDF format. The application also allows to run Picoting using Max2, which we won't do here, but I will explain it later. And the two final steps are really for biological interpretation. And this is firstly differential analysis, which can be done one versus all or comparing exclusively two samples or two clusters. And we can, for example, select two samples and run the analysis. This will give us the regions that are enriched or depleted in our mark of choice. And based on these regions, we retrieve the genes, and we can run a classic gene set analysis using the MSIG-DB database. And we can visualize which are the top gene sets that are depleted or enriched in, for example, H3K27 ME3 mark. And we can now, on the UMAP, plot the signal of H3K27 ME3 on the single cells. And also plot the average of some of the enriched pathways on any given clusters. So I hope this was not too quick and gave you a good overview of all of the features of ChromSCape. So now I will share again my screen. So I want just to come back to a few of the features to give you a bit more details. And I want to stress out here that ChromSCape can take either featured data format, which is dense or sparse matrix, but it can also take raw formats and process it into any kind of features. It can be genomic beans, peaks, or genes, as I told you, but you could also give any annotation using a bed file. And for the visualization, I'm giving here an example of this mark, and we are using the same cells, analyzing this mark using either peaks, which is more supervised, and the corresponding CPM normalization, or small beans, in which case we would prefer using TF-IDF normalization. And this gives us quite different results and can be two different approach to analyze our datasets. And these plots were actually made using the application and saved as a PDF format. Now that you have your cells and you can define clusters, and based on these clusters, as I showed in the application, create the coverage tracks. And just to give you the details, we retrieve the cluster information for each single cell, and going back to the raw read, we produce big WIG files and upload them directly into the application. So based on this clustering, we can also do the peak calling, as I told you before. And what is the main point of the peak calling? So there are two goals. The first one is to filter the gene to feature annotation. And as you can see, if we use large genomic beans or large peaks, there might be several genes in the vicinity of genomic beans. But using peak calling, we can safely detect where the signal is enriched. And here, the gene A is really not really enriched in signal in this bin, so we can remove this gene link with this bin. And the second and most interesting point is to create a consensus peak annotation. And using this consensus peak annotation, that was called on each of the rare or not rare population that we identified, we can rerun the analysis in an iterative manner to have a more supervised approach, but with this time peaks that were called on the single cell clusters, which would be better than calling on the bulk population, which would miss some rare peaks. So this was the use of peak calling. And now I would like to give you 3 other package features that weren't mentioned in the application. So the 2 first are just to say that you can use ChromSCape by scripting, and this is basically for reproducibility and to also run multiple analyses on parallel on your high performance computing, for instance. And also that ChromSCape creates a really organized file structure, and this file structure is recognized automatically by the application and allows to load automatically all the analyses that you did, and specifically all the thresholds that you selected. And you can really benchmark several thresholds and several cluster choice, and this can be shared by simply sending the folder. The second point that I didn't mention, but which is linked with what I said before, is that we can analyze from the raw data multiple angles on the same cells, and this is using the single cell experiment package and specifically the alternative experiments. So here one user might be interested in analyzing, for instance, single cell attack-seq using 5K base pair bins. This will give unsupervised dimensionality reduction and clustering, from which the user can call peaks and rerun on the same exact cells the analysis to have a more fine-grained clustering. And using this fine-grained clustering, the user can finally count on gene body or gene promoters to have easier biological interpretation. And this is really what we wanted to implement in ChromSCape, this iterative approach. And finally, the package has multiple features to help with the user experience. So I won't mention them all, but for instance, we implemented browser cookies to remember where your main folder is located, so you don't have to select it each time you open the application, or a helper that guide the user when they are a bit lost in the buttons of the application. So now that I described hopefully all of the features of the application, I will give 2 examples of use case. And the first one is using a single cell cut and tag of the mouse brain that was published by Bartosovich and colleagues this year. So we reanalyzed their data, and the experimental design was that the authors dissected mouse brain and sequenced different histone marks, either active, such as H3K4-ME3, or repressive marks, such as H3K27-ME3. And the authors basically wanted to know if it was possible to distinguish different cell types of the mouse brain, this complex tissue, simply using epigenetic marks. So using our package, we have done the answer by dimensionality reduction, and we can identify 8 clusters quite easily, which if we retrieve the cell type annotation from the published paper, matches quite well the clusters. So this is interesting because using repressive mark, we can see that all of the cell types have quite different landscapes. And how the authors did to really see which cluster was which cell type is by looking at marker genes. And for example, ODIC2 is a known marker genes in brain development. And we can see when we plot the signal of H3K27ME3 at the vicinity of these genes, that the gene is mostly repressed in the lower cluster, while it's not repressed in this cluster and might therefore be expressed. And we can do the same thing for NeuroT2, which is expressed in all the clusters except the lower one, which is a neuronal cluster. And we can do the same once again for H3K27 acetylation, which is an active mark specifically enriched at enhancers. And here, once again, we find more or less the same annotation with some discrepancies. So now that we saw that we can identify cell types using simply epigenetic single cell, we can wonder if we can, from our single cell types, identify stresses. And in our lab, we're particularly interested in the breast cancer chemo resistance. So we used single cell ChIP-seq to analyze the H3K27ME3 landscape of breast cancer cells upon treatment of chemotherapy. So we used the triple negative breast cancer cell line MDI-MB468 and treated them with 5-FU, which is a second line treatment used with patients. And what we observed after a couple weeks or more of this treatment is that only a very small fraction of the initial population managed to survive, and these cells were not able to grow under treatment. And that's why we call them persister cells. But after more weeks of treatment, some of these cells gained the ability to grow under treatment. And we call them these cells the resistant tumor cells that are the cause of relapse in breast cancer in patients. And we really wanted to see the dynamics of the histone, the repressive histone mark under this treatment. And this is what we obtained from this dataset. Basically, we found 4 clusters, and if we go into the detail, we can see in the upper part of the UMAP that the 3 gray chemo-naive, which are the tumor cells that didn't see any treatment, are grouped in 2 clusters. The persister cells are grouping together with one of the resistant sample, and the 2 last resistant samples are together. And there are many multiple interesting things in this graph, and the first one is that the persister and resistant cells are quite different from the chemo-naive cells using in the repressive landscape. So a lot is happening here in terms of epigenomic landscape. And the second interesting thing is that the 2 persister states are grouping together, and that might be indicating of a common state, which could be a bottleneck and could be, for instance, treated with epigenomic drug or other drugs to prevent them to become fully resistant. So we can, in the first time, we can wonder what is driving the strong difference between the chemo-naive and persister cells. And to do so, we can do differential analysis and look at the coverage of the samples using ChromSCape, for instance. And we can see at the vicinity of interesting genes driving epithelial to mesenchymal transition, for example, TGF-beta1 or FOXQ1, that the repressive mark is lost at this loci, which would enable expression of such gene. And this was confirmed by single-cell RNA, or some other interesting genes that are drug transporters, for instance, are de-repressed. So this was interesting, and another question we could ask is that we observe quite a strong heterogeneity in the chemo-naive population, and we can wonder if this heterogeneity is linked somehow to the persistent state. And the first thing we can look at is the cell-to-cell correlation score of any cells with cells of the persister yellow cluster. And we can see that the the pink cluster is significantly more correlated to the persister cluster than the C4 gray cluster. And going back once again to the coverage tracks, we can see that some genes are already de-repressed in the pink cluster, and this de-repression is kept in the persister state. And this might indicate that the cells of C2 are more prone to express the persisting expression program. So hopefully I showed you that ChromSCape is able to run a full-on analysis of single-cell epigenomic datasets, and particularly that it can use a wide variety of datasets thanks to the ability of counting onto different features, and to be quite flexible in terms of parameter customization. And finally, it can be used in several biological contexts, such as cancer or cellular development and differentiation. And in terms of perspective, we really want to implement multimodal analysis, which is, for instance, per tag, but I'm sure there will be others, expression and histone in the same cells, to add more biological interpretation, and of course to optimize resource and reactivity, for example, to handle very large datasets. So thank you very much, and thank you for inviting me, and if you have any question, I would gladly answer them.
All right, thank you, Pacome, for the nice presentation. Let's give you some round of applause.
Thank you.
People enjoying it, and it's so much fun. All right, so let me bring one question to the stage, and
Yes.
you can address it. I can broaden up this question about do you have plans to include UMAP in addition to t-SNE? So it's basically about visualizing high-dimensional
Yes.
data in low dimension. Can you talk a little bit more how you decide on the number of clusters when you visualize the data? So perhaps start with the question and then.
Okay. Yes. So yes. So basically, in the first version of the application, we only implemented the t-SNE approach, which was really used in most of the analysis. But now this t-SNE is optional, and we automatically run the UMAP. So the UMAP is in the new version of the application, and you can optionally run the t-SNE, but this takes longer to run. And to answer to your question, Michael, the clustering is done using, so we calculate the Pearson correlation between cells, and we use this as a similarity metric, and use this as input for the hierarchical clustering. So you can either run simple hierarchical clustering, which could be quite not robust, but you could also run consensus clustering. And this is using a Bioconductor package, which is consensus cluster plus. But it is resource heavy. So It depends on the size of your datasets. But this can help you detect the right number of clusters.
All right, wonderful. So I don't see many other questions. So let me ask, you showed on your slide, there are many different technologies about single-cell epigenomics. So can ChromSCape analyze, like, do the integrative analysis, like handle data from multiple technologies?
So basically, at the moment, ChromSCape can only analyze single modal datasets and not, for example, pair tag, which multimodal and integrates both RNA and histone modification in the same cells. But we want to be able to analyze both modalities of a single cell in the next release of the application. But you could, of course, analyze simply single cell attacks from multimodal experiments using ChromSCape, for instance. But you would have to use other package for RNA analysis, for instance.
But in principle, if you have like two data sets, ChromSCape can correct for batch effect.
So right? Yes, we use yes. So if you speak about multiple like different experiments, we can of course integrate them, and we use fast MNN batch correction, which actually works quite well for single cell epigenomic datasets. So yes, you can use this kind of integration, but not not yet for multimodal datasets. Yes.
Well, lots of work for future.
Yes, lots of work.
All right. If there are no other questions, let's give our speaker another round of applause. And
thank you very much.
Thank you all for attending.
Thank you very much.
Enjoy the rest of the conference and see you next session.
Thank you. Thank you. Bye-bye.